Hello, we're going to study cellular respiration today. Cellular respiration is the process in which organisms obtain energy from food. Anaerobic and aerobic respiration are the only ways energy is obtained for cellular function and life processes. When the supply of energy is completely cut off, it basically means death. When it's disrupted, it leads to very serious health conditions. Therefore, understanding the nature of cellular respiration becomes very important. Germinating and non-germinating seeds. A plastic bottle as respiration chamber. A carbon dioxide gas sensor. A USB link interface to connect your sensor into your computer. On your computer, open the Pasco Capstone data collection software. Pasco has many other softwares that you can use, but I'm going to use Capstone today. Connect your USB link into your computer. Connect the cables to your carbon dioxide sensor and connect it to your USB link. As soon as you do this, a green light will start flashing on your carbon dioxide gas sensor. I'm going to calibrate the carbon dioxide gas sensor by pushing on this button until the flashing green light turns into a solid light. And then I will release it. It will stay solid light, and after a while it will start flashing again. Flashing light will mean that it has completed calibration. While your sensor is calibrating, you prepare your first setup with non-germinating seeds. I use pea seeds in this case. 25 pea seeds that are not germinating. Empty them in your respiration chamber. When your sensor is calibrated, lay it on its side. Respiration chamber. Lay the respiration chamber on its side and plug your carbon dioxide gas sensor into it. If you let it alone, it will tip over on its side. So I will just move it towards my computer. Support my carbon dioxide gas sensor with my computer. On the Capstone software, select a layout with table and a graph. Select your measurements as time and carbon dioxide concentration. Select your measurements on your graph too. Set up your recording conditions. We want data collection for 10 minutes. So click on this and set up stop conditions, time-based, and 10 minutes is 600 seconds. So set it up to 600 seconds, click OK. And as soon as you click on record button, it will start data collection. And I will change the frequency at which software collects data. I will set it to take a sample every 15 seconds by clicking on this down button. It is set up to 15 seconds right now. And I will hit record button. It will start data collection and it will continue for 10 minutes. Now data collection is complete for non-germinating seeds, and you don't see much change in the graph because of its scale. It ranges from 500 to 5,000. It's a huge scale. So we're going to auto scale this first by clicking on this. And you see this clear trend. You see a lot of fluctuations because of the random motion of the carbon dioxide gas through the sensor. Sometimes it passes more, sometimes passes less due to its random nature. But you see the trend that carbon dioxide level increases as non-germinating seeds respire at a very very low rate because they are dormant right now. In order to calculate the rate for carbon dioxide production, which is the measure of rate of cell respiration, you are going to create a best fit line for this, a linear fit, by clicking on this button where it says curve fit. Select a linear fit for your data. And the rate for cell respiration in non-germinating pea seeds in this trial was 0.0596 parts per million per minute, per second, I'm sorry. Now we can go ahead and try germinating seeds and calculate their rate of cell respiration. You remove your carbon dioxide gas sensor from the respiration chamber. I have 2 of these bottles right now. If you have only one for your experimental setup, remove all the seeds. And since carbon dioxide gas is heavy, it settles on the bottom of this container. So you either tip it over and wave it to get rid of the excess carbon dioxide, or simply just wash it. But I'm going to use a new bottle for my second trial. I'm going to get my pea seeds that are germinating, 25 pea seeds, and I will empty them in my respiration chamber. I will lay it on its side and plug my carbon dioxide sensor into it. I will wait for a minute or so my carbon dioxide gas sensor adjusts itself to the container, and then I will collect data. Start data collection. Go to your passcode capstone. It holds the data from previous trial. I'm going to click record, and it will start a new trial. Click on record, and it starts data collection right now. It will continue for 10 minutes and 600 seconds in this case. It will meet to 10 minutes. Now data collection is complete for germinating seeds. I'm going to remove carbon dioxide sensor from the respiration chamber. Now as you can see in the screen that carbon dioxide level has changed from 600 to 2000. It was a quite change. So I'm going to calculate the rate of cellular respiration again by performing a best fit line. Here, linear regression, and here you see the slope of this line is 2.36. So rate of cellular respiration, you would say 2.36 parts per million per second. Now I'm going to plot both data on a spreadsheet to make comparisons between the two sets. Well, of course, doing multiple trials will allow you to give a degree of confidence on your decisions, but for the sake of this video, we're going to just compare single trials from both groups. And I will show you how can you see both runs. This is the second run with germinating seeds, and when you go click on this button, you can select between runs and you can find the first run, and in the graph by going on this button and clicking on this, you can select the first run with non-germinating seeds, and you can transfer values for cellular respiration from your graph into your data table here. This is what I created. Since we're comparing rate of cell respiration in germinating and non-germinating seeds, I have created a table like this. And you actually want to determine the rate of respiration per gram of living tissue. So I have masked these pea seeds before we do the trials, and germinating seeds weighed 5.5 grams and non-germinating seeds were 5.1 grams. So I'm going to divide whatever the value that I calculated in the capstone for rate of cell respiration and divide it by the mass I get from seeds, and it will give me rate of cell respiration per gram of living tissue. So I'm going to transfer the values here. For non-germinating seeds, it was 0.0596. I will do 2 decimal points, so I will round it to 0.06. 0.06. And for germinating seeds, I will switch between my runs. I will switch to run 2, and slope was 2.36, and I transferred that value here, 2.36. And it's an obvious difference between germinating and non-germinating seeds, but doing multiple trials would allow you to assign a degree of confidence in deciding that difference is not due to chance. So this is not adjusted values. I'm going to do adjusted to mass, let me say grams of living tissue. I'm going to calculate the adjusted grams of living tissue values for rate of cell respiration by dividing cell respiration rate of cell respiration I calculated in the Pasco capstone software and divided by this number, which is the mass of the seeds. And I do it by entering an equation here, equal rate of respiration divided by mass of germinating seeds. And I will bring this to a 2 decimal point. And for non-germinating seeds, it is equal this number divided by mass of non-germinating seeds. And I will bring this to, again, 2 decimal point. Now I can make a graph to visualize my data. I select my 2 groups and I select their adjusted values, and I select insert chart, and I want, since I'm comparing 2 groups, I want a bar chart, columns, non-germinating and germinating seeds. I will hit insert, and that's my chart. I can change the chart title to rate of comparison of rate of cellular respiration in germinating and non-germinating pea seeds. That could be my chart title. I'm going to change the appearance of my graph by putting in labels for these 2 groups. And update. I'm going to insert axes titles. Horizontal is germinating and non-germinating seeds, and vertical is rate of cellular respiration per grams of living tissue, and unit for this is parts per million per seconds per grams, and label for vertical axis is rate of cellular respiration per grams of living tissue, and unit is parts per million per seconds per grams. And update. This is how my chart looks like right now. It is easier to understand than understanding this table. So this is the visual representation of my graph. You can easily see the difference between the cellular respiration rate of germinating and non-germinating seeds in my chart. As I said before, you can assign degree of confidence to your decisions when you're saying rate of cellular respiration in germinating seeds is significantly different than cellular respiration rate in non-germinating seeds, but it would require you to make multiple trials and perform statistical analysis. This video has presented a protocol in which rate of cellular respiration can be measured. Many other investigations that deal with rate of cellular respiration can be designed by modifying this protocol.